All right, good morning, everyone. Wanted to at least just start off by thanking you for attending this morning. I know it is very early in the morning, the last day of the conference, so I appreciate the effort to be here, and hopefully it will be worthwhile from that perspective. So, as you said, my name is Tim Nieters. I am the Senior VP Strategic Portfolio Development for CPC Scientific. CPC is a peptide and oligo CDMO with facilities in China as well as soon to be in the US. I've been in the industry now about 30 plus years. And one of the things I've always enjoyed about the peptide industry is one, just the sheer breadth of products, right, and targets that peptides can be used in, but also that kind of constant innovation and development that utilize the peptides. And that's part of what I'm speaking about today, which is peptide receptor radionuclide therapies. That's a mouthful, so I will just refer to that as PRRT or even RT throughout this presentation. But essentially, the PRRT is using a targeting peptide to a specific receptor that is overexpressed on a cancer cell, and then delivering a cytotoxic payload to kill that cancer cell. As always, I think one of the neat things with peptide developments or developments in general for drug therapies, you tend to have either one significant discovery that launches into a therapy, or you have a long history of innovation that's building on top of each other. And so radionuclides obviously have a long history, but from a drug therapy standpoint, you really didn't see it until most recently. Even if you go back to the discovery of somatostatin and then eventual analogs of octreotide that were synthesized, discovered in 1982, you still didn't really have a PRRT therapy approval in the US until the kind of the mid-2000 teens, if you will. And so on the right-hand side, you see really over the past 6 years, several FDA-approved PRRTs. And these are those examples just in a structure form with the brand name. You can kind of see a couple different peptides, a couple of different metal isotopes, and chelating agents that are used, or that are approved. And I'm going to get into some of those details here as part of this talk. So why peptides in PRRT? Everybody's had those discussions about how peptides vary, are different from small molecules, or how they differ from large molecules. But there are some very important benefits of peptides as they relate to PRRTs. And so obviously the first one is the smaller size, right? Compared to proteins and larger molecules, they enable that rapid penetration to the target cancer tissue. You do have the positive pharmacokinetics, right? With the high concentration in the target tissue region, as well as the rapid renal clearance of the peptide. Peptides also have the high affinity in nanomolar ranges, right, with a specificity towards a kind of a wide range of receptors in the cancer cells. And then we'll call it kind of malleability, if you will, the conformation structure of peptides can be both rigid and flexible, tend to lend themselves very well to this sort of PRRT where you're trying to lock in with the receptor of these cancer cells. And then, of course, you have the general, right? They're less immunogenic than larger molecules. They are generally non-toxic as compared to small molecules, and then minimal side effects, hopefully from less off-targeting affinities or binding. And this just really kind of lists that in a table format, right? Comparing the different molecules, peptides versus larger, kind of walking through the benefits or the cons of each one. I think one thing to just really kind of point out again is that elimination through the kidneys. So that rapid elimination, especially if you're dealing with a radionuclide through the kidneys, not getting to the liver, but also then, which something I hadn't mentioned was the manufacturing cost associated with peptides as compared to larger molecules. So there are some very good benefits of peptides in this sort of therapy. This slide just really kind of alliterates the kidney filtration and why peptides tend to be a little bit more beneficial. Again, due to their size and their overall shape, they're able to be cleared through the kidney. Charge is really kind of a wash. You have that with both molecules. But then again, that malleability, that flexibility with the conformation of peptides as compared to larger molecules allow for that to be eliminated through the kidneys and not making it to the liver. So another important part of PRRTs is the metal isotopes that are used. And whether you're designing an application for a therapy, a diagnostic, a theradiagnostic, you're going to consider different particle waves, whether it be alpha, beta, and those are really kind of based on the type of, the range of tissue penetration that you're looking for. And then you can see some very commonly used isotopes associated with those metals. This slide kind of, again, demonstrates that. So the purpose of the RT, right, the radionucleotide, is to destroy the cancer cell by directing energy into those cells of the tissue without obviously harming the healthy cells around it. And again, just that demonstration of what you might be trying to accomplish with the particle emission, whether you're alpha, beta, auger, the penetration that you're looking for within those cancer cells will determine the type of therapy that you would develop. As anything, when you're developing a therapeutic, you're going to walk through some general major steps in that development cycle as you're preparing for a clinical trial to get into humans. That obviously starts with cancer cell with the target identification. So what is that specific receptor that you are looking for? Then that peptide identification, so what is that synthetic peptide that has that high affinity for that receptor target? And then the chelate design, right? So designing that synthetic process for the manufacturing of the bifunctional chelate with the linker, and doing that in such a way that that chelate molecule does not interact or affect the binding of the peptide to the receptor. And then from there, you would choose, as I was showing the radionuclide, what are you trying to do? What kind of penetration do you need? What metal isotope? Developing that radiolabeling procedure, and then obviously moving then forward with the other preclinical studies, tox studies that you would need to set up that kind of final dosage. And so from CPC's perspective, we're involved in the beginning aspects, kind of 2 and 3 of that synthetic for the peptide and the chelate design. These are some of the different bifunctional chelates that we have worked with. You're probably familiar with most of these. DOTA being a kind of a little older one, DOTAGA, I'm going to get into a little bit more detail in this case study. We'll mention one of the newer ones. Again, as you always see, innovation is the trap. This molecule, this chelate is able to, it's a little bit broader stability across pH ranges. And so having that ability is, we all know that in cancer cells, cancer areas, it tends to be a little bit more acidic. So the trap offers a little bit more stability across that. These are some of the peptides that are overexpressed in tumors. Again, I think this list is fairly common. Most people have heard of these peptides, whether it be octreotide, substance P, and then some of the indications and where they are expressed in the different tissues. And so for this, for this part, we're talking about, these are some of the other PRRT peptides that we have synthesized. You can kind of see, again, the receptor that we're targeting, the actual peptide, and to some extent the chelate that we're using. And we have done quite a bit of work in here. But for this particular case study, I'm going to focus on the GMP process for the DOTAGA-labeled urea-based peptide PSMA inhibitor. And so just to provide a little bit of background on that, prostate-specific membrane antigen, or PSMA, is a metalloenzyme, about 750 amino acids in length, that is expressed in the prostate. It is found in a couple of the other tissues. But the advantages of the PSMA, maybe over other cell markers for prostate cancer, are one, it tends to be expressed even to 1,000-fold higher levels than what you would find in healthy tissues. It is expressed in all stages of the disease, so it is very effective from that standpoint. And the conformational structure of PSMA allows for an improved internalization of the therapeutic or the payload that you're delivering. As you're designing these, there are different types of peptide-based inhibitors with a C-terminal glutamate that bind to that active site. The 3 major ones that are used are phosphorus-based, thiol-based, and urea-based ligands. The first two have some limitations. Phosphorus-based, based on its high polarity, can be difficult, limits the penetration of the blood-brain barrier. Thiol-based, again with oxidation, can be unstable and probably not good application for clinical usage. And then you do have urea-based ligands, because of their high binding affinity and stability, make for very good use. And this is what we tend to see more in the PSMA area. That negatively charged linker can help reduce off-target retention. And then the hydrophobic aromatic structures incorporated into that linker can really improve the PSMA binding while also decreasing the kidney uptake, or said the other way, the more rapid removal from the kidney. So there are some certain things to consider, important critical factors for a urea-based inhibitor. One is the charge, as we mentioned, the chemical components, the linker, and then the hydrophobicity. And so for the linkage composition, you're really looking to incorporate amino acids, and in most cases, you're gonna use the D amino acids to increase the metabolic stability of the linker. In any case, you need a structure that would not contain a enzyme cleavable bond. I also mentioned the hydrophobicity, and so you do want the multiple aromatic rings because they do improve that interaction at the S1 accessory pocket. I'll show you that in a little bit. But again, to be able to deliver your payload, internalize that into the thing. This is a neat drawing of the active binding site. So essentially you have I went back there. Essentially you have a 20 angstrom entrance funnel, and you can kind of see how you have to get down into that pocket. So that glutamate pocket is where the urea would connect. You also have the arginine patch. And then we talked a little bit about the hydrophobicity and those aromatic structures, and that's at that S1 accessory site to be able to deliver that payload. So again, that's where that kind of design of the peptide, the receptor, the chelate, the radionuclide all come together as a therapeutic product for prostate cancer. So again, for this particular case study, as I mentioned, it's going to be for the DOTAGA-labeled Urea-Based Peptide PSMA inhibitor. That's really comprised of those three components. So you have the PSMA binding urea moiety, the D amino acid linker, and the DOTAGA for the radionuclide chelating. We tried a couple different process development strategies from a synthesis standpoint. This was the unprotected fragment condensation approach, where we obviously start with the 2-CTC resin, add in the lysine, continuing with the D amino acid couplings, also then finishing with the DOTAGA coupling, and then a cleavage and deprotection step to isolate that intermediate. And then from there, doing a fragment condensation liquid phase coupling with the urea, following with another deprotection step to yield the product that we're looking for. There were some disadvantages to this particular process, mainly being the overall crude yield and the crude purity from this step. So a second synthetic process that we tried was a fragment condensation, but using DDE protection. And again, starting with the 2-CTC resin, having the D-lysine with the DDE, following the same kind of process coupling step with the D amino acids and the DOTAGA, doing that DDE deprotection on the resin, and then obviously the urea coupling and cleavage again. We did see an increase in the overall yield for this particular process, but crude purity still remained low. So we continued with some of that synthetic development work, and again tried a more standard FMOC TBU synthetic route. You can kind of see where this was added in a couple fragments. So you have the glutamolyurea lysine added to the CTC resin, another fragment followed by the two D amino acids, and then also the DOTAGA coupling at last, and then doing a final cleavage, deprotection and cleavage step to isolate the final product. And this particular one had a significant increase in overall crude yield and crude purity, and through purification steps, a very high final product purity and this is what was used for this particular process. I know I'm not showing a lot from a cleavage and purification standpoint in terms of the process. I don't want to minimize that. That's actually a very important component of this, and where a lot of process development work was done. Everybody's aware of some of the issues with chelating agents during the cleavage step, the formation of chiral isomers, and so doing that analytical work along with the process development synthetic work was a key part of this to be able to make a very pure crude and then ultimately a very pure peptide. And this just kind of summarized the three different approaches we took, again with a very good yield from crude yield and crude purity, resulting in a final product purity of 99.3. I know you can't see that from this slide, but also a very high purity from a chiral standpoint. And so this process took about 10 months, which is a very tight, aggressive timeline to get from initial start of a project all the way through the IND filing and the CMC dossier prepped. Again, I can't stress enough, the success of this project was really in that first initial 3 months, doing concurrently that analytical method development along with that process development to develop a process that would deliver a high purity, allowing us to move very quickly through the pilot batch, the tox batch, and the GMP batch, ultimately to hit that filing in 10 months. So a very successful development process for us. I think a lot of it was based on some of our know-how and work that we had done prior to this to be able to achieve that in a 10-month time frame. We have worked with a lot of other DOTAGA chelates, and we have citations that can be referenced. We're obviously happy to talk through any if you're interested in any of those. And then we have done quite a bit of other PRRT work and have multiple citations and development work that we've done. And so we're obviously happy to have those conversations if anyone's interested. But again, a very successful project based on the history of the work that we had done. I'm going to switch gears for just a second and kind of move over to how we've taken some of that conjugation chemistry and some of our background and moved into POCs or peptide oligoconjugates. And so CPC, having a long history, now 20 plus years in the peptide industry, solid phase and solution phase chemistries, that conjugation experience that I was kind of talking through in organic synthesis and the oligos that we've added over the past several years has all led to kind of a strength in these peptide conjugated moieties, and in this case, oligos. We have in-house analytical capabilities for both the peptides and the oligos, scale-up capacity to work with customers as they work through discovery, very quickly through discovery, and then scale up for clinical and commercial. And then, of course, obviously that global quality standard that's required to file, whether it's in the US or with any other regulatory agency and customers' requirements. So again, just to kind of highlight those discovery platform, we tend to work with customers who are looking through a numerous amount of peptides and oligos, what will deliver best, what they're trying to deliver from an oligo standpoint. So we do have an integrated peptide platform, again, very large scale if needed, but small scale with very quick turnaround to work through these, and obviously with a lot of different modifications from a peptide standpoint. And again, on the oligo side, a lot of different technology and ability to turn those around quickly to be able to do this conjugation work for discovery in a very quick time frame to allow for a lead candidate to be identified and move forward from a development standpoint. This is our overall oligo. I'm not going to get into this. I just wanted to kind of share this with you. But we do have Sherry Ambry is here this week. Would love to talk with you if you want to have discussions about any of these, any of our offerings, and what we can do from an oligo standpoint. With that, I'm just going to do a very quick team introduction. If you're familiar with CPC Scientific, you're probably familiar with Sean Lee, who founded the company back in 2001. And then Teresa Chang is our president here in the US of CPC Scientific. And I just want to mention some of the team members that are also here this week. Howard Huang is our Senior VP of Business Development. Sherry Ambry, I mentioned earlier, is our oligo specialist. Then we have two very talented BD folks, Andy Yan and Michael Jung, who are here representing the East and West Coast operations. And one last introduction, or at least announcement from is Chris Gothard. He is the person responsible for putting this presentation together, working with the CPC team. And so just want to thank you to him for putting that together. With that, I'm going to kind of stop. I'm not going to go through the rest of the slides and just say thank you. And if there's any questions. It's a long walk to the microphone.
Yes. And a very tall microphone. Hi, Tim. Very nice talk. Thank you. I have a question on the regulatory side. Mm-hmm. This kind of molecules, there's only like 2 or 3 amino acids in there, right? How they will be regulated if it's going to be regulated as small molecules, how would you recommend to your clients in terms of the impurity control?
It's a good question. I don't know that I'm the best person to answer that question, though, for you. So obviously, you've seen quite a bit of approvals over the past 6 years, right, with those particular. I think in this case, you'd obviously have to have a very high purity peptide, right, which is where kind of what we're focusing on. But I don't know. I'm happy to put you in touch with somebody who can kind of talk through a little bit more of that regulatory aspect from an impurity standpoint.
Thank you.
So.
Hi.
Hi.
Do you actually perform the isotope chelating step in your facility?
No, we are not involved with the radiolabeling aspect of that.
Do you work with your partner is there a partner that you work with?
We have, a lot of times the client that we're working with who's interested has that partner because they've identified the radionuclide that they want to use, and they work with that radiolabeling procedure from that standpoint. We're really delivering the peptide and the linker.
Okay, great.
Yeah, very nice talk.
Thank you.
Enjoyed. So I'm wondering, can you comment on your PSMA, process, the urea part?
Unfortunately, I can't expand on that, but I will say from the proprietary standpoint, it's not a specific reagent or technology that only CPC has. It's more the know-how to help reduce those impurities, increase overall yield and purity of the crude, and then those purification and analytical technologies. So it's not a specific reagent that's proprietary, but I just can't share that process that we used and developed for those PRRTs.
Okay, thanks.
Mm-hmm.
Hi, Tim. Thanks for the presentation. A short question on the CMC side. Do you see the need of a very tight control of your, after including the DOTAGA, for example, on residual metals in the solvents, for example?
I think with what you're delivering, I don't think that there's going to be a significant tight control in that regard. So obviously you're working with a radionuclide, from that standpoint. So that's really where some of the more off-targeting and the radionuclide is more of what you're concerned with from a toxicity standpoint.
All right, but you don't see that as a huge concern?
No.
Okay.
No.
Thank you.
And again, if the process development done correctly, from that standpoint, you eliminate a lot of those concerns. And it tends to be a more standard specification for our peptide.
Sorry, I have another question. Is for this particular compound, what's the highest, impurity?
Highest impurity?
Yeah, in this process.
I don't remember what the highest impurity was. But again, in 99.3, you can imagine it was a very pure product. There wasn't a very large single impurity associated with this. You can kind of see from the chromatogram, right, that there was really nothing else. So very low levels of other impurities.
Okay, great, thanks.